everyone. Welcome to the 9th webinar in the Data Science webinar series organized by Manav Human Atlas Initiative. Myself, Chalish Kumar, moderator of today's talk. And as you all know that the total allotted time for today's webinar is 1 hour, out of which talk will be of nearly 40 to 45 minutes. Then we will have a question answer session of about 15 minutes. I request all participants to use QA tab at the bottom of your screen to put the queries. I repeat again, use only the QA tab for putting your queries. You can put your queries in between the talk that will be answered by speaker during the question answer session only. Now, I would like to introduce Dr. Amit Vutha, speaker of today's webinar. Dr. Amit is currently working as a lecturer, Department of Mathematics, the Ohio State University, USA. He has completed his bachelor's as BSc Honors Mathematics from St. Stephen's College, University of Delhi, and completed his PhD in Mathematics from Ohio State University in 2013. As far as his experience is concerned, Dr. Amit had served reputed institute in various capacity. Like he worked as a lecturer at Ohio State University from 2014 to 15, worked as a postdoctoral researcher at NCDS TIFR Bangalore from 2015 to 16. He worked as a postdoctoral researcher at ICTS TIFR Bangalore 2016 to 18. He also served as assistant professor at Ajin Premji University, Bangalore in 2018. He later joined as a lecturer again to the Ohio State University in 2019 and working till date. His research interests includes mathematical biology, dynamical system, and game theory. So without taking too much of time, now over to Dr. Amit for his talk on the network matters. So over to Dr. Amit.
Yeah. Thank you very much. Thank you for this opportunity to speak. And again, this is a topic which is maybe very important because of everything that's happening now, even though academically the current situation is very, very difficult and complicated. But hopefully in this talk, I'll present some ideas which are different from traditional ways in which people think about epidemics and disease, and hopefully if you're interested in taking these ideas forward, there may be some things to work here. So going forward, if at any point you have questions or in the future you would like to contact me, you're very welcome to do that, because especially with the current scenario, we're all stuck trying to find answers, and it's not easy to do. Okay, so let's start. The plan for my talk today is for me to talk about three main pieces. The first piece, which I'm going to say, is the role of connectivity, right? So maybe if I have to add something here, systems which are connected in different ways give different outcomes. So the first main point that I want to establish is that if you think of a disease, and if you think of a disease spreading on the system, again, it does not have to be a disease. It could be any process that's spreading. It could be a rumor. It could be some information. This is part of a theory which people call as contagions, that there is some kind of a spread. Every time there's any kind of a spread, the exact domain on which you study the system becomes very important. On one extreme, you can say everything is connected to everything. There's lots of different objects. Whereas on the other extreme, you have each piece of the system is separate from every other piece. So every piece behaves like its own subsystem, and then in general, right? Any real world problem that you study is going to be in between these two extremes, between being fully connected and fully isolated. And so that has some impact. Okay. The next thing is I want to talk a little bit about existing work on uh, how people study epidemics and networks. Again, it's a very well-established field. People have been studying this for a long time. There's lots of very good work and things I've referred to at the end. So there's plenty of material, but I want to say a few things about it, and then of course finally talk mostly about a couple of recent and alternative approaches to thinking about epidemiology in general using data, right? Because that is the goal of a seminar such as this, that we are living in times when using data to solve problems is very powerful, and doing this the correct way can have a lot of impact and can change how you tackle certain problems. Okay, we want to see how using data that is available in a certain setting can help you, uh, and what kind of tools there are. I will introduce some of these ideas as we do that. So this is the plan. So let's start. I guess the first big question to ask is why exactly should connectivity matter, right? What is the if I'm studying a system, why is it important for me to understand how the different pieces are connected? And again, this is something which is true for any system, not just for diseases. If you want to study the human body, if you want to study something else, there is some value to exploring this idea. Okay. So very quickly, just focusing on epidemics, because that is what we're going to be doing today. The first thing I want to point out is if you take any epidemic in history, right? And you look at the early phases. Okay, so I just want to point out that we are interested in what is happening in the early phases. Most epidemics will have this general pattern where they will start and have some growth, but you don't know what happens after that. Okay. Some epidemics will go down, some epidemics will live longer, some epidemics may go up. So there's different possibilities in some sense. Is very quick growth of cases, and so understanding something about it is useful, and having an estimate which works as you proceed forward in time, so that maybe time this way and cases this way, has some value for us. Okay, why is every epidemic growing exponentially? What is the reason for that? The second point I want to make is any study that you have, right? You have an epidemic, it's happening live in front of you. You want to, for example, if you think of COVID-19 right now, everyone is interested in knowing what is the rate at which it's spreading, when is the peak, when does this epidemic die down. But any prediction you make, okay, so this is point number 2 that I want to make, is that estimation of parameters, right, which is generally a goal for any epidemiologist. You want to describe the system as closely as you can according to some model. The problem with this is contingent on data that starts with a very small amount, okay, that is initially small and evolves rapidly. Okay, so I can write down a model, I can run some estimation algorithm, but then tomorrow we're going to have more cases. Thursday there will be more cases, Friday there will be more cases. And so anything that I have is going to change, and as it's changing, the amount of data that's available is also changing. Okay, so somehow that makes it more complicated, and so we have to be careful about that. The last point, of course, is again with real world epidemics. this is always a problem, right? That your populations are always changing. So if you think of most governmental responses to the current pandemic, it involved, so I'm going to say, lots of different factors here, but maybe something like responses of authorities. Right? So if you are a government, you say everyone has to stay in the house, that has some impact. If you say everyone has to go back home, that has some impact. If you say only people of certain age can move, that has some impact. If you say people have to wear masks, that has some impact. So when we are studying the system and you're studying the spread, again, I like to think of this, using some chemical analogy, right? So you have this beaker, and in this beaker, we want to model what's happening. Okay, but this beaker itself is changing as we're studying the problem, and so you need something that's a little bit more dynamic, something that where the number of people who could be infected, the number of people who can infect other people, all of these factors are changing, and that complicates the problem. And that's the first thing. The second thing is there is some kind of connection between some important factors, right? So there's the spread of disease. This is somehow linked to movement of people, and I think that's what I was saying before. And that is somehow linked to whatever your authority does. Okay. And then you have some extraneous factors. You have some temperature. You have the geography of the place. You have how people are living in some areas. Some areas have dense clustering. Some areas people live far from each other. So all of these factors somehow make the problem very high dimensional. Okay? So my conclusion is that real world epidemics are extremely high dimensional from a modeling, from a theoretical standpoint. Extremely high dimensional. And so as someone who wants to study the problem, you have to really have a very good idea of saying, I'm ignoring these factors. I'm assuming these factors, right? What advantages does that give me? On the one hand, it might simplify the problem, might give me some tools, might give me some ways in which I can compute stuff. But on the other hand, every assumption that you make is going to simplify the problem and lose some detail of the real world, right? And so because of that, there are some issues. Right? So this is the first point I wanted to make. So with that in mind, and again, I want to show you some of these classical definitions. I know maybe Dr. Goyal a couple of weeks ago gave a very detailed and good talk on this. But classically, right? So this is the classical idea approach to finding this number called R0. Okay, so this is what's called our reproduction ratio. And the idea with the reproduction ratio is you're saying, look, there is some population. Draw this picture. For now, let's assume it's a box. We don't know what's happening in the box, but initially there is no disease, right? So fully susceptible. And then if I come from the outside and introduce one individual with a disease, okay, so one diseased individual, and then I say initially, what kind of rate will I have when that person comes in? How many people will that one person infect in this population? That is telling me something about what the reproduction ratio is. And again, this idea was, even when they introduced it, it was a simple idea. It's somewhat problematic because populations are never fixed. If you take a city like Pune or Bangalore, we have very large populations, right? So you can say, look, this box is fine. I can bring one person with a disease and see how fast this disease is spreading, and that's going to give me some number. But again, the city is not homogeneous. Okay, so first of all, the number of people in the city are changing. The way people are distributed in the city is changing. Someone living in an area with high density is going to have a different outcome than someone in low density. And so this idea is useful, but only to a point. So that's the first thing. There's a more kind of modern idea, right? And this is maybe another, I want to say, more recent and time sensitive version of reproduction ratio. Okay, so this is called RT. RT again, you can think of it just like what we were saying in terms of the reproduction rate. But now we are saying, look, okay, when someone comes in to the population initially, so we had this picture before, so let's go back and get this. But this is just the snapshot. This is just day one. On day two, what can happen is this person might have 3 people. And so the rate that this person has could be different from the rate that that person has, and could be different from the rate that that person has. So each individual can have their own rate at which they're infecting people when they're infected. But along with that, there's also some time sensitivity to it. If you think of day 5, individual 1 may have a higher rate at which they're infecting someone than day 7. Okay, so a most general version of a reproductive ratio should in fact, on the one hand, it's an average, but the question is, what is it averaging? Are you averaging across time? Okay, so I guess the point I want to make is every reproduction ratio is an average. Okay, but then it's an average on a system which is changing with space, but system changes spatially, it changes temporally, and it also has some dynamic aspect to the populations inside. Okay, so reproduction ratios are useful numbers and they're averages, but within the idea of the fact that it's an average is for us to remember at every point that, hey, this is a system which is changing, and we are only doing this on average. Okay, and because of that, there are some problems. Okay, so that is the first main point. Okay, so very quickly, we've talked about some of this before, but I want to highlight this again. We want to say what are potential sources of problems. Okay, so specifically with regard to epidemics and modeling them on networks, why can this lead to some complications? So the first thing I want to say is this total, right? Which is what we assume is fixed when we have certain kinds of models. Okay, this is never fixed. Okay, changes. So maybe we should be a bit more specific here. Distributed in lots of different ways. And because of this, right, you have to be very careful when you say this is R0 for this population. Okay, so what if you have a large city? So let's go back to our pictures. Here's my large city. But within the large city, and this is true for any city, you can see this, say there's some big center, there's some other center, and there's some other center. Okay? And then in between, there are some connecting parts. So maybe I'll draw some arrows like that, and some arrows like that. But just this is a cartoon of some city, right? Of a closed population. Okay. So the first question to then ask is, if I'm averaging the rate at which this disease is spreading across all individuals, what am I gaining and what am I losing by not recognizing the fact that there's a lot of clustering, right? That somehow this unit seems to have its own life, this unit seems to have its own life, and this unit seems to have its own life. Is it advantageous for me to say, okay, this is almost like there's only three different things spreading to each other? Is that a good approach? Do I have to take each circle as a separate closed system and write some model for it and treat that as a fixed total? So this idea of a fixed total itself is problematic. Okay? So you want to be careful with that. The second thing that's genuinely a problem with most epidemiology is the duration of infections. Okay? So again, if you look at the current understanding of most diseases, when people describe how long am I going to be sick when I have a certain disease, right? So again, you can think of COVID. It is something that's current and something that we're seeing in front of us. This quantity itself is not very well defined. Okay, so for example, most diseases have a distribution of infection times. Okay, so one of the kind of theories that's there for a disease like COVID is, hey, actually, there is two types of COVID, right? We don't know if this is true, but this is what people are saying currently, that there's one type of person who would spread it very fast. There's another type of person that would spread it very slowly. Okay, so going back to my city again, so let's do that. And I'm going to take this multiple colors guy. Right. But then what you're saying is, look, yes, it's spreading, but actually there's two arrows. Okay. So there's one arrow like that, another arrow like that. The black is some kind of slow spread. The red is some kind of fast spread. And the rate at which the fast spread and slow spread happens between these different units in my system is different. What impact does that have on the overall disease, right? So that's one question. You can model that. But more generally, what impact does that have on my estimate of the spread of disease? Okay, so if I'm saying R0 is 2.1, is it 2.1 for everyone as an average? What if 99 out of 100 people in that system are not spreading it at 2.1? They're only spreading it at 0.1. And there's two people who are spreading it at 8.7, right? So is that average actually telling me something useful, or is that average actually misrepresenting the system in some way? So this is going into using average statistics in general and why it's problematic. But yes, this is as a fact, most diseases have this. Okay? COVID most certainly has it. People say there's a 2-week window where you have to stay home if you have the disease. Within those 2 weeks, you should not touch anyone or speak to anyone, or something like that. But we know that that's not the case. There's been multiple instances of spreads happening in later time, earlier time. Okay, the age of the people involved matters. The viral load of the person involved matters. So it's not a very simple detail. Okay, so you want to be careful about that. And then the final part, which again I think is something that is true for any statistical system. Okay, averages only give you one level of information. Okay, so if I take, I teach a class, I have 100 students. I look at exam 1 and I say on average the class performance was 78, right? But then if I look at the actual way in which the scores are distributed, a lot of people got very low scores and maybe some people got very high scores. My average is going to be somewhere there. Okay, but for me to actually understand how my class is performing, I have to recognize that, look, there's one group of students who are doing much better than average. There's one group of students who are doing much worse than average, right? The average tells you something about overall in a snapshot. If I stand 200 meters away from the scores and look at it, it's telling me some information about it. But then really things like variance or higher moments, which are again statistical estimates of a system, um, tell you a lot more about the system. Okay, and again, if you go to the kind of picture that I had here, my model city, there will be some problem with actually just averaging things the way as it is. Okay? So if the red sub-city is bigger than the yellow sub-city, you want to average those numbers while respecting that information. Okay? So none of this is to say that look, there's exact answers to these, but as we read publicly available information about what's going on, we always want to remember that every time someone talks about these ratios, these are in fact averages. Okay? And because of that, they are problematic. They're useful to the point. And in fact, the way we have defined it before, maybe I should say that as a final comment. It's not that averages are useless, okay, but we want to say generally R0 is the upper bound of lots of different ways to think of the reproduction ratio. Okay, but again, even this statement is somewhat misleading because you could say that R0 of a system is 4, but there may be a part in the system where the R0 is 9 or something, right? And it's not going to reflect that. It's not going to reflect that when we compute it. Okay, so because of this, I guess the point I'm trying to make is that in general, right, you could have completely different systems. I can give you 10 different cities or 10 different networks. You can choose thousands, the same number of people in each one of those systems, okay? But they can give you the exact same reproduction ratios, right? If you plot the number of cases versus time, when you look at that data, you will see the exact same plot for each of these objects, okay? So I can run one system where I say this disease only spreads among men. I can run another system where I say this disease only spreads among people who have 3 friends or more. I could run some simulation where I say this disease only spreads with people who are less than 5 years old, right? And the truth is, you can find enough examples where if you just look at the output of the system, right? So many different possibilities exist for something that gives you one of these plots. Okay, and so when someone comes and says, "Hey, this is the plot for my region. This is the reproduction rate. This is what R naught is. Right now, R naught is less than 7, so the peak is next month, and the curve has been flattened because we did something else." That kind of information is useful till a point. Okay, but beyond a point, it is not respecting the detail and diversity that is inside your system, and I'm going to say lots of different possibilities. Can give you the exact same plot. Okay, so exact same case reproduction, exact same curve, exact same peak, and the reason why we see these peaks and these kind of exponential curves in the first place has got more to do with the fact that if I know a certain number of people on average, and you know a certain number of people on average, and we're both doing something that affects people that we know, then you get an exponential process. Okay? So something being exponential is not that surprising. If you forward a post on WhatsApp to 3 of your friends, and they forward 3 of their friends, and you get the plot of that, that will look exactly like this shape that we see for most epidemics, right? So there is some value in it, but the value in it has to be qualified within what we have. Okay. So with that in mind, quick check of the time. So yeah, maybe go a little faster now. So we're talking about epidemics and networks. Okay. Networks are mathematicians like to call them as graphs. but people like to call them networks. Generally, what is a graph or a network? I'm going to say any collection of two groups of things, right? On the one hand, you have things called nodes. On the other hand, you have things called edges. Okay. Just like what I'm talking before, the nodes are just the different circles in your system. Okay, so those are nodes. And edges are things that connect these nodes. Okay, so in this case, I have an edge there and I have an edge there. So within this framework, you can see there's a lot of different diversity in how you can study problems. You can look at large networks with lots of people connected in a certain way. Okay, so every time you study a problem that involves connectivity, you can frame it as a network problem and has a lot of potential. Generally, the questions you want to ask with any epidemic or network type of process is, how do you want to model nodes? Right? Should I model nodes? So I'm going to say some examples of this, of epidemics, epidemic models on networks. Okay. So how do I choose my different nodes? What are the different ways to do that? One way you can do that is, each individual is a node. You can treat subpopulations as nodes. Okay, you can treat geographic regions as nodes. Etc. Right? So there's so many different ways to do it, and people have done that. If you go in the literature and look for what are the different ways in which people have used networks, people have done lots of different things, and all of these are valid and have something useful that they're telling us. Okay? But more generally, the question always comes down to, how do you want to represent the population that you have? Okay, so is every individual in my system the same? Okay, I guess. What you want to say is, are there subclasses within the population, right? So, for example, with COVID-19, people always talk about people above 60 are more at danger, and somehow they have to be careful. People who have certain immune conditions are more likely to fall sick. It's true, right? So should we model that separately, right? Children who go to school are less likely to get infected, but more likely to spread the disease or something, right? So there's all these kind of questions that come about. And in general, again, this is boiling down to the idea that when you think of populations in general, there's a lot of ways in which a group of people are different from each other. They're similar from each other. And the more accurate an answer you can get for that from the point of view of your disease, the more useful your modeling efforts can be. Okay. The other ways people have done this is sometimes people use these things called multiplex networks. So maybe you have 2 layers, and then I have the same network. So maybe I'll go with that shape. Okay. At one level, I can think of how people are sharing information. Let's say, actually, that people are, let's call it the contact network, meaning people, how they know each other. So this person knows these two know this third person, and this person also knows this person. Okay, something like that. But then you may have something else. Okay, so you may have information in your system. In other words, me knowing about the disease affects everyone else in some way, right? So you can think of the government as one of these nodes. If the government knows that a certain area has a certain disease, it may communicate that to each node separately. And so these are examples of what are called multiplex networks. You have the same nodes, but you have multiple layers of connectivity. And somehow the interplay between these different layers, right, has some effect on how the disease spreads. Okay, so within each network problem, these are standard considerations that you have to ask, and depending on the level at which you want to study the problem, you have to choose different nodes, different edges, and different ways in which you're representing the population that you have. Okay? And so just to give you an example, a city like Bangalore, where I'm from, if you have to do something at the level of a small apartment, right, apartment complex, or maybe a small village on the outskirts of Bangalore, you will have a different way in which you'll represent the nodes, and a different way in which you'll represent the edges. Okay, maybe people will be the nodes, edges will be who lives in the immediate family. But if you want to model the entire city as a whole, you could treat each ward as a node. Okay, you could treat the flow of people between these different nodes as an edge. Uh, you could do some other stuff which says, hey, wards which have more people have some higher density or something like that. Okay? So there's different scales, and what scale you choose has some impact with this. Okay, so here's an example of a simulation that again I've linked to the code for something like this. It's at the end, but I just want to point out. So here's an example of what I was saying earlier. Okay, so this is 3 different architectures. Okay, and again, you can kind of choose the parameters for these different architectures in a way so that you get the exact same plot. I've just not done that just to give you some contrast in which you can see it. So you can see they're described here: truncated power law, Erdős-Renyi, and homogeneous. These labels are telling you something about the nature of connections. Okay. And so, but what's happened on each of these is, again, someone has plotted the time on the x-axis. Right? So going this way. This is just the cases on the y-axis. And you can see, if you actually look at how these different networks are connected, a power law network assumes that each person on average has some, so if you look at node degrees, right? Node degrees, right? So if I look at, say, I think this was for 1,000 nodes or something like that. If you look at node degrees in the truncated power law, what you're saying is there is some kind of a drop like that. It's a very small number of people. So I'm going to say number of nodes, right? So on a power law, you get this structure where I think it's dropping quite rapidly, like 1 over x to the power of n for some n. So x to the power of minus k. And as you change k, you can get different power laws, right? Erdős-Renyi is something that's completely different. Okay? You're saying there's some probabilistic way in which 2 nodes are linked. Okay, so people like to call these as random networks. They're not exactly random, but they're random in the sense that there is a probability that I'm linked to you if you're both sitting in the same system. So something like if you live in a again, if you live in a small community, there's a probability that you know another neighbor in your complex, right, or something like that. Okay? And then finally, homogeneous is saying that same level of connectivity for all nodes. Okay, so what's fascinating is, yes, I mean this version of the graph doesn't show that, but you can actually choose these 3 completely different architectures to give you the same end result. Okay? So that is something to keep in mind. A couple of other things that I quickly want to mention because we are moving ahead on time. Generally, when people study epidemics and networks, and again, there's a lot of really good work on it. One approach, which is kind of more common with people who comes from statistics, right? And maybe even physics, so especially a statistical physics approach of understanding networks involves writing down things called master equations. Okay. So master equation is me saying on average, what is the certain quantity doing? Okay. So of and the work of Boltzmann, Boltzmann was able to show that there's some distribution of energies, right, of kinetic energies, that not all molecules are uh, ha- uh, have the same level of kinetic energy. Some of them are slower, some of them are faster, but there's some distribution. Okay, so this process of taking a microscopic system and arriving at an average description. There's a lot of work of this sort with epidemics and networks. A lot of it is very useful. Some of it is not very useful, but people have done some good stuff. Okay, and again, there are some links at the end of this talk where I've linked to this work. So there's that line of work. It's useful and interesting. There are more. I would say works well for large networks. Okay, so just want to point out. For large networks, this approach is great. When you think of small networks, right, or I'm going to say highly structured networks, there have been these alternative approaches. So again, I've just given you some keywords here. You can look for them if you want. Essentially, the idea is that is there a way in which network structure is repeated, okay, across the network. So what we're saying is, if you take most tissue or most biological networks, right, and you study, you look at the network, like you have some picture or some visualization. What people find with a lot of networks found, especially in living systems, is that large parts of the network look very similar to other parts. Okay, so if I have some network which tells me how all the proteins in some gene are impacting each other as they produce some product, right? You will see that on average, a typical protein connects with certain other proteins in almost the same pattern, and that pattern is repeated in a lot of different places. Okay, this word called motifs was very common. People use this a lot. This kind of work is picking up a lot with epidemics on networks. Instead of studying a very large system, if I study a very small system which has a lot of structure, meaning if I know what a system like this does, and I know what a system like this does, and I know what a system like this does. Okay, so these well-established small systems which have high levels of similarity and self-similarity. Can I somehow combine my understanding of these to maybe understand a system which looks like the following? Okay, so what I'm going to do is I'm going to take this pentagon, and within this pentagon, on each node, I'm going to put a triangle. So something like that, something like that, something like that, and something like that. Okay. So if I look at this object on the right, it has some symmetry. It does not have the full symmetry of the objects on the left. Okay. Within each node, there is some similarity in which how it connects to other guys on the pentagon and to two other nodes, right? And the fact that there is a basic repeating unit. Okay. Like this unit is over here and here and here. There's been some recent tools which help you transfer what you can understand about dynamics of these simpler objects on the left to these guys. And when you have one of these networks, you can somehow go back down and shrink it. Okay? And so that's where ideas like lumping and symmetries have been very useful. Okay, so I don't want to spend too much time on it, but these are very new and interesting ideas. So here's an example. So you can see this is a generally a large network.
Sorry to interrupt, Dr. Amit.
Yeah.
We have 5 minutes. It's
okay.
yeah.
Fine. So maybe I'll just go a little faster. Thank you, Shailesh. So this is a large network, and you can see with the way someone has colored this, they've associated certain nodes to be similar to each other. So you can see the yellow nodes and the blue nodes and the orange nodes all have similar connectivity. So you can go from here to here, right? And then finally, if I shrink down all the yellow guys as one of these, and all the blue guys is just one node, you can shrink the original dynamics, which is very large, to a much smaller network. Okay? And so these kind of tools have been fairly common. Okay, I do want to get to the main point of today's talk, which is to say that we can use data that's available to say something about the spread of diseases. Okay, so in general, there are always questions that you should ask. What data is available? Typically, with any problem which you can study using network epidemiology, you may not have full data. Okay, so I'll just make a quick comment. Generally, only partial information is available, right? Partial data exists. Okay, so because of that, any method that you have should be robust to that and should respect the fact that we only know certain things about this. Okay. What assumptions can you make about the underlying system? In fact, how is the disease transmitting? Does it have a certain spread rate? How are people moving? Okay. And in terms of the connectivity, really what you're saying is how do different nodes impact each other. Okay. And so the closer you can get to answering these questions, the more accurate your simulation can be. And in fact, the more accurate the results of the simulation can help you to decide what you want going from. Okay. So with that in mind, maybe I'll just highlight some of these papers that I was going to present. Number one is a very old paper, right? This is from 2001. Okay, so there was a foot and mouth epidemic that occurred in the UK, right? And as this was happening, people, started getting news reports which said that on Tuesday there's a farm where some cattle were tested. On Friday there's a farm where another where another cow had foot and mouth. On next Tuesday there's an incidence of foot and mouth in some other farm. So if you think of the map of the entire country, right? Again, UK. I'm just going to draw a cartoon. There are some nodes. Okay. On day 0, you have something like this. But maybe on day 1, this node tested. On day 2. Some other node tested, etc. Okay, so you have some network level information of you have some kind of spatial information about where the disease is coming up. So how does networks actually help us estimate something here? Okay, so what they did in the study, and again you can look it up. You can see on the left here. This is in fact a estimated RT. Maybe on this axis versus time, right? And so the way people use the networks is the following. The algorithm is to say that, look, suppose I have these incidences happening on certain days. One way, a very simplistic way in which I can see who is linked to who is suppose this guy ended up with disease on day 8, right? What I then want to do is I want to say, what are all the possible places where I could have gotten this disease on day 8? Okay, so I'll construct a network like that. On day 1, right? On day 2, this guy got the disease, so it's possible that the disease arrived this way on one of these guys. Okay, so what you can start then doing is start with a system which is so I guess if I have to describe this very quickly because of time, I'm going to say construct all possible parent networks for a given infection. Okay. Repeat for all infections and all days. Okay, and again, you can make certain assumptions. If you know that foot and mouth can only travel by a certain amount, right? You can incorporate that so that the guy on the top does not affect the guy on the bottom. Okay, If you do this for all infections in all possible networks and then just average, okay, what you would then do is in fact obtain some kind of idea of how this reproduction rate is depending on a specific node. Okay, so that's what's happened here. You can see the red, the blue, the green, and the yellow are in fact probabilistic estimates of how the reproduction rates are changing. And in fact, there was one node which had an overall reproduction rate, which was persistent throughout the whole process. Okay, and in order for us to prevent a future epidemic, it would be very useful to think of that node as being what is controlling the what is most likely to impact other nodes in the system, right? One quick example and I'll stop. So here's something that's much more recent, again, done in the same way. Okay, so this is using what's called Bayes' theorem. Okay, so what happened here is people, very similar to the foot and mouth disease, people have done this with Ebola, right? If you look at Ebola as a disease that's spreading, and I have some network of all the towns in this region, and I just have this incidence data, what I can do is with every incidence, I can construct all possible parents and just average, okay? And the way Bayes' theorem works is if A and B are two possibly connected events, right? What Bayes' theorem does is it helps you connect P of A given B with P of B given A. Okay, so in other words, if I know that in Mumbai, you're more likely to have rain on the west side of the city, right? If it rained, what is the probability that the rain happened on the west side of the city? So these are, again, probabilistic ways of asking questions. But in practice, in simulation, the best way in which you can do this is to just run as many simulations as possible. These are what are called Markov chain Monte Carlo methods. Okay, so if I run 10,000 simulations of all possible networks that could result in the numbers data that I see in front of me, and then I just average on that data, right? While I'm constructing these networks, I assume that these people are more likely to be connected than something very far away than those guys. Then somehow that estimate that I have is going to be more accurate for me to do that. Okay, so maybe I'll stop in the interest of time. Yeah, I mentioned this in some ways. Just to summarize, the spread of diseases on non-homogeneous, okay, meaning networks which have a lot of structure and where there's a lot of interconnections, is very, very different from assuming that you have thousands of particles and they're all similar to each other. So we want to keep that with us with every bit of data that someone gives us over the next few days. Okay. Question number two, point number two is that reproduction ratios are averages. Because they're averages, there are problems with it. We want to be aware that there are these problems. Doesn't mean they're useless, but it only means that when you have a certain system which is small and very structured, that average is only going to tell you certain things and not everything you need. Okay. And then number three, I've kind of started talking about it, data-driven approaches. Okay, just counting how many different ways you can have a certain outcome and treating that as a proxy for what the rate should be at that point is much more powerful than me trying to fit some high dimensional model saying that, hey, I have 7 equations, I have 14 parameters, I have some optimization method, I'm computing some trajectory. In some sense, you lose more information that way. You're much better just running something many times on some simple structured assumptions and treating that as a proxy for what you want. Okay, so I'm going to stop here. Here are some very quick details. This book, in fact, is fantastic. And most codes, so I'm going to say plenty of codes are freely available. So if you can run Python and just look for this book, almost every model that's used in the book is available online. You'll be able to download it, run your own code, choose your own network, introduce your own assumptions about how you want your network to spread, and the program will do the work for you. Okay? So it exists. Link number two, I think is very, very good because it highlights some of the more mathematical details, right? Because the mathematics of averaging on these structured domains itself is complicated. Okay? People don't know everything they need to know about it. Epi model, this is an R package. So I'm gonna, if you don't like Python and you like R, Epimodel is very good. I've used it and very useful for us. And finally, this Ebola paper, which is the one that I linked to at the end. Okay, very useful. In fact, I would say example of locating some of these ideas like hotspots, centers, super spreaders. Okay, which you can do yourself by just counting how many times you have a certain outcome versus not having a certain outcome. Okay, so I will stop here. Thank you, and then if you have questions.
Yeah, thank you, Dr. Amit. Uh, because of constraint of time, we have limited questions here. So I will put few of them, like which are means more towards the Indian perspective of this epidemic.
Sure,
One
I'll do
one
my
is
best.
that what is the possibility of second wave
So
in your view? Yeah.
yeah, so my point of view is, I guess the question itself is it's so it's okay. So let me answer it this way. It's defined assuming that there was a first wave which stopped and there's a second wave which is starting, right? So I guess when you say is there a second wave, you're assuming that something like this will happen. And in my view personally, and this is not based on some deep study of the Indian situation, is just that there's really statistically no difference between that and something like this. Okay, so what you're perceiving as a first wave and a second wave, in fact, might be just a single wave. Of course, you may say, should I be trapped in the house forever once I leave outside? I mean, there are human reasons why this question is useful, and I understand. But I think mathematically, uh, there may be no second wave because there was no first wave. So
So in the similar way, means in the line of the previous question, there is another question that can the curve have oscillatory behavior?
yes, the curve can
When
very
so
definitely have oscillatory behavior. And in fact,
okay,
most epidemics, if you see, don't just stop. If you look at tuberculosis in India, there's in fact some kind of persistent level. Uh, can you have the same up and down swings like what you had now? It's a very complicated question to answer because this disease is very new, and a lot of things that many governments did was also very new, right? We have never done anything like this before. So can there be a very large jump and a fall, and again a very large jump? Uh, I think that this structure is more likely, but again, this is only my personal opinion,
okay.
uh, and not based on.
Thank you,
Yeah.
Dr. Amit. One more question, one last question. What you say means there is a question that objects the count means mortality rate and number of count in India's perspective. So
Okay.
what you say, ki, according to the model,
Uh,
are they correct, or is there any question mark on the stats which is provided? Means if there
uh,
is, yeah, this is difficult question, but yeah,
it's a difficult question because I feel I mean, most authorities or governments are trying to do the best that they
yeah,
can, right? So I think rather than the data being incorrect, I think it will be a function of how developed the regions are. So maybe Mumbai and Delhi because they're big cities, but also because there may be more testing or there may be more hospitals may give you a different count. Uh, I wouldn't say the numbers are wrong, but I would just say they will depend on how you collect the data. So if you collect the data differently, you can get different numbers. But this is not unique to COVID because every disease has this. But
yeah,
I think one of the advantages of these data-driven approaches is it doesn't matter what whether how someone tested or something. So these models are very close to the data, right? So tomorrow, if you come and say I have more data, then you can rerun the model and get a better estimate. But yeah, I don't think anyone is lying or presenting false information.
yeah, sure. So one small last question that came from one person
Hmm.
that is related to is during the population count we are considering the unborn individuals or not epidemic? This is only simple question.
Sorry, can you please repeat that?
Yeah, are we considering unborn individuals for considering the population while modeling epidemic?
I say unborn,
Yeah,
children who are not born. Uh,
yeah,
that's a good question, but I guess my point is, why would you worry about unborn people? So generally, I guess, so let me answer it this way. In classical models, like SIR models, which have differential equations, there is a birth rate and death rate. These models will assume
okay,
that over time, babies are born at some rate. But yeah, something that's more data driven and more local. If I'm studying something that happens over 6 months, maybe the unborn population is not so important. So
yeah.
that's maybe what I would say. But yeah, it's a very good question.
Yeah. Thanks, Dr. Amit, for this interactive and interesting talk. And literally, that modeling of network is very important for epidemic thing, understanding the epidemiology in depth. So, thanks, and we will put the questions which are still left unanswered to you for
Okay.
later answers. So, I request all participants to fill their feedback form for today's talk. And thank you all for joining us for this webinar. Really, and I really thank Dr. Amit for sparing time from his schedule for this talk. And I would also like to introduce the participant that in the series of the webinar, we will have Professor Ajit Kembavi from Ayuka, Pune for the next webinar on 22nd of July. So he will be delivering his talk on machine learning and deep learning with application to astrology and biology. That will be really interesting topic attend. So for further details, you can visit our social media platform like Twitter, Facebook, and Insta. And that will help you to get to know about this webinar more. So thank you all. And I also like to thank outreach team to organize this webinar and whole technical team for their support. Thank you all and all the best. Have a good day.
Thank you.
Thank you. Thank you, Dr. Amit. Thanks.
Thank you
Bye.
very much.